So what is DNA methylation? DNA methylation involves the transferring of a methyl group from a donor S-adenomethionine molecule onto a cytosine base, forming 5-methylcytosine. In cells, this is an epigenetic process which helps repress and regulate the expression of genes by preventing the binding of transcription factors. The concept of methylation was first discovered in the late 1940s by Rollin Hotchkiss when he was able to detect 5-methylcytosine in calf thymus. From this discovery, researchers later realized that majority of methylation occurred on cytosine bases that were connected to a guanosine base, forming a CpG dinucleotide. In 1975, Arthur D. Riggs also established that methylation was an epigenetic mechanism, meaning that methylation did not alter the DNA sequence directly. As mentioned earlier, methylation works by methylating a cytosine base, and which is typically attached to a guanosine, forming a CpG site. This transfer is mediated by a family of DNA methyltransferases. The addition of a methyl group prevents some key transcription factors to be able to bind to the DNA sequence, which then prevents its transcription of the gene sequence. There are two types of methyltransferases that are involved in methylation. Maintenance methyltransferases, such as DNMT1, is responsible for copying the methylation pattern from a mother strand to a daughter strand, so that gene expression remains identical after DNA replication. When methylation patterns need to be changed, however, de novo methylation is needed. DNMT3A and 3B carry out this role, where they are responsible for methylating unmethylated cytosines, thus making a new methylation pattern and altering gene expression. De novo methylation often occurs during development, such as gametogenesis or embryonic development. Methylation plays a key role in genomic stability and integrity. For example, DNA methylation is also used in other epigenetic modifications, such as histone modification. Through DNA methylation and other processes, the association of DNA to histones can be altered, which changes how easily transcription can occur. DNMTs will bind to enzymes that regulate histone modification to make DNA pack more tightly to the histone, thus making transcription harder. On the other hand, if DNMTs were to malfunction, faulty methylation or lack of methylation could occur, which would result in faulty or unwanted gene expression. This is the case with cancer, as a direct link has been discovered between malfunctioning DNMTs and the development of cancer.
Created using Powtoon.